components of the case, and the figure show all the components in the case will be those regions, and one dilution place and one ELISA place which coded the ACE2 protein. And here is the CE certificate we already get, and some validate, like in Indonesia we already done. So here is the manual protocol of how this process can be done. So because the ELISA method can be done manually, and we can see here, so the whole process can be finished within 2 hours. So within 2 hours, at least 94 samples or 92 samples can be tested. So this is a really high throughput one. Besides the manual method, actually this ELISA kit can also be done automatically. So Vazyme SVNT case is compatible with various type of automatic ELISA workstation to achieve the automatic analysis. With the high throughput workstation, at least 720 specimens can be tested within 2.5 hours. So if we have the analyzer, the process can be reduced very easily. So I can just like click and check. So we just need to prepare all the samples and put them into the analyzer, and we press the start button and wait for the results. So here, the manual, all the manual process will simplify to dilute the washing buffer and dilute some regions and pull all the regions into the container and pull the plates and the tubes test samples into the machine and start the program and waiting for the results. So here is the software, how the analysis operating system looks like. And this is the final report. So after testing, actually we can have the reports like this. So this will show the barcode of the patient's sample, and show the OD or the signal of the testing, and show the positive or negative at the interpreted results. So how the result interpreted, no matter we use machine or the manually, actually is all based on this same formula. So for the quality control, the NC or negative OD control should be over 2.8, and the positive control OD should be below 0.2, because it's a competitive method. So if the more neutralizing antibody in the samples, that the signal will be much lower. So if there's no antibodies or the negative sample, the signal will be higher. So based on this inhibition formula, we can get an inhibition number as a different percentage of the inhibition. If the inhibition is over 20%, that means that is a positive one, indicate the presence of the RBD neutralizing antibody is detected. But if the result is below 20%, that means this is a negative sample. The negative sample indicates the neutralizing antibody is absent or below the detection limit. So how about the LOD? We use the monoclonal neutralizing antibody that diluted in the negative serum sample to mimic the positive neutralizing antibody serum samples. And the limit of detection was evaluated as over 92.5 nanogram per milliliter. Then the antibody can be detected by this ELISA kit. So in the reaction mixture, if the final concentration of the antibody is over 4.6 nanogram per milliliter, then the antibody can be detected. So this method is really sensitive. So how about compared to the live virus neutralization test? So 84 cases of the positive serum from the vaccine subjects with the brain or the live virus analysis titer over 4 was identified by this kit, with the brain 50 titer ranging from 4 to 4 to 24, 24 to 48, and the titer increasing in the brain 50. The median inhibition rate also increased from 5% to 26%, at least to the 75%. So the result of the ELISA analysis is quite correlated to the brain experiments. So here is a summary of the analysis. So the sensitivity of this kit is 96.55, and the specificity is 100%. for these 52 samples which identified as negative in the brain experiments. So have because as we all know the gene script have so many publications in to study to compare with the brain 50. And so how about this method compare with the gene script SVNT products? So from here we can see the clinical collected specimens was tested on both gene script ELISA kit and Vazyme kit. The correlation between these two kits was analyzed and the R square is 0.294. So actually these two methods quite correlated. And and also compared to the brain 50. So if the lab cannot achieve the brain 50, I think these measures can be a very good solution to test the neutralizing antibody. So here is the validation we done in Indonesia. So as you can see here, our products have very high sensitivity even at the very early stage of the COVID-19 diseases. So as we see here, we have the like total 90% sensitivity in the 108 samples, and the accuracy for 225 sample is 93.8. So except to reduce the qualitative assays, actually this method can also easily achieve the semi-quantitative assays, which means we can give a titer. So to evaluate the titer of neutralizing antibody between samples and compare the efficiency of the vaccines, the semi-quantitative assay is very important and very easily achieved by the SVNT. So the method is just we need to full diluted each samples, and so to check at which dilution fold the negative is transferred from the negative positive sample to the negative sample. The dilution fold for that sample will be as titer. So with the titer data, we can compare the analyzed neutralizing antibody level between different samples. And for so we can also come with the group of the many enough data. We can also compare between group to group between the different vaccine platforms. So we can tell which vaccine have the better efficiency to help us, or after in vaccination, which vaccine can help us to get a high level neutralizing antibody to help us to and protect the fight against the virus. So I think for no matter for the research or people all around the world, the most concern is how long the neutralizing antibody can last or how long can persist in our bodies. So it is very important to know the antibody, especially the neutralizing antibody kinetics in the circulation system after vaccination. So the SVNT can be used at the very beginning of pre the vaccination, and after vaccination, we can continually test until at which time point the antibody is already dropped to a very low level, and at that time, I think it will be a good time to take the reboost of the vaccination. So as the conclusion, the SVNT ELISA kit is user-friendly and can be easily performed with microplate reader. And compared to the live virus or pseudovirus analysis, the ELISA is time-saving and high-throughput assay, which can be significantly boosted by using an automatic ELISA workstation. And beside the qualitative detection, SVNT can be also used for the semi-quantitative assay, which is an ideal tool to the antibody kinetic assay in the pre and post vaccination study. And this is the brief, all the presentation for today. And I'm very thank you for your time to listen, for your attention to let me briefly go through the neutralizing antibody test. Thank you. That's all.